Wait till it does that bit. So yeah, so good morning everybody. For those who don't know me, my name is Paul Shaw, researcher here at IBH in Dundee. And it's my great pleasure today to introduce today our two speakers for today's seminar, a topic that's very close to some of our hearts here in Dundee, titled Understanding Plant Germplasm Collections, Data Sharing and Breeding for Climate Change. So today's talk will be presented by Michael Kantar and Anna McCormick from the University of Hawaii in Manoa, who you'll see here. So Michael is an associate professor. His work focuses on plant breeding, neo-domestication, CWRs, relatives, and food security. And Anna is a postdoctoral researcher whose work focuses on exploring genetic diversity and species distribution, including developing new breeding strategies for adapting crops to climate change. So without further ado, I'll shut up and hand the floor over to Michael and Anna.
Well, thanks so much, Paul, for that great introduction, and thanks so much for having us here. We're really excited to talk to you about the stuff that we've been doing for the past couple of years with these germplasm collections. So, Anna, next slide. So everybody eats, right? This is something that seems obvious, but I think is very much exemplified by my friend Cookie Monster. But every culture has their own favorite plants. So I love this map that Colin Curry made about a decade ago, showing all the different crops and all their Pavlovian centers of origin, looking at multiple domestications. And what you can really see from it, I know it's a little small on the screen there, but what you can see is that every different region of the world has developed this technology of domestication, and every single culture has their own favorite plants, whether it's Van Gogh's sunflowers that we're seeing in the bottom left there, or if it's these beautiful woodcut prints in Japan of millet, or it's a watermelon from the Renaissance, right? We all choose our favorite plants and we decide to eat them and focus on them. Yeah, next slide, Anna. But one of the things that's really important is, where's all this genetic diversity? Where is it all stored? And what we really what has happened is, over the past decade, there have been great studies exploring how well we've been doing in conserving our global germplasm resources. And so what you see in that table there is a great paper from Peterson Wambugu that goes over all of the genetic resources in the world and where they're all stored. A lot of you are probably familiar with the GeneSys platform. You can look up all of these accessions there. But in total in the globe, we have about 7 million accessions that are actually stored. A much lower number are actually available in about 1,600 gene banks globally. And what you're seeing on the infographic on the left and the right are these wonderful infographics that have been generated by the National Plant Germplasm System in the US that are freely downloadable, that show how gene banks and conservation work. And really, they go through all of the steps and they show why we should really be caring, they show how we store basically all of these genetic resources and where you can get them. Next slide, Anna. So the lab here, we really care about these collections because of climate change. And you can see that I like maps. There's something that we like making quite a bit in the lab, but this map is meant to be incredibly depressing, and I hope it's depressing you guys right now. Nothing like a nice, morning bout of depression, I guess, or for Anna and me, an evening bout. What you're seeing here is an additive index of climate hazards. So in this case, if you're white, climate is not going to change by 2100 in that particular grid cell. The redder you are, the more additive cumulative hazards that you have in that particular grid cell. If you're blue, your climate is getting better overall relative to these hazards of things like food security, acidification, heat waves, storms, and Camilo Mora put this index together. Basically, the reason I say it's depressing is our best case scenario in this map is no change. And almost every part of the globe is multiple cumulative hazards. And this is solely looking at it in an additive way. We're not looking at any interactions or anything like that, solely in these additive hazards that have been documented. And so we really care about the collections because we know that that's where a lot of this genetic diversity is stored. Next slide, Anna. So this is why you should care. I'm sure you guys already do because you signed up to listen to us talk about them. But and I know not everyone's a big fan of the Tanksley and McCooch figure from the 90s, but I think it really does a really good job, and it's why we care so much about the wild relatives and landraces, or these early domesticates. The genetic bottleneck that occurred means that we lost a lot of diversity. And a lot of that diversity that was lost was not helpful for our agroecosystems. But when we talk about breeding for climate change and we talk about our germplasm system, one of the things that's really, really helpful is that in the wild species and the early domesticates, they were subjected to thousands of generations of natural selection in very often very extreme environments. And so those alleles, in this case, that are represented by the different colored squares, we have access to those and that history of evolution in the wild species and land races. But because plant breeders have been so good at selection, we've lost a lot of those in our modern lines because we do a much better of managing our agricultural systems to make the plants happy than a lot of the a lot of the regular parts of the species distributions. Next slide. So one of the things that's really, really tough is we have 7 million accessions. How have we gone about trying to choose what accession you want to use? There have been a lot of different ways. So one of the most popular ways has been something that's called FIGS. It's the focused identification of germplasm strategy. I'm sure many of you are familiar with it. This is a great paper by Halp and Schmid from about 5 years ago that showed how to do it. And in this case, what we're really doing is we're looking at the univariate distribution of, if we're talking about climate change, of the environmental variable, say heat, and we're taking the extremes. If we want to deal with heat, we're taking the extreme edge of that distribution of that environmental variable, and we're taking an accession that is, again, a landrace or a wild species that exists in that extreme range, and then we're choosing that as a parent. This has a lot of drawbacks. It tells us nothing about the genetic relationships with any other line, and it's also just taking that univariate distribution. The second way that has been also really, really popular for the last 40 years are core collections, and particularly the mini core collections, where instead of dealing with our tens of thousands of accessions, we're dealing with our usually two to 300 accessions that are represented that through stratified sampling represent 80 to 90% of the total genetic diversity of the collection. This makes it tractable, but in this case, then we don't really necessarily have as much information about where they fit in the distribution of all of our species and all of our accessions. Another very popular method has been that folks at Kew Gardens have done a lot of work on is phylorelatives. So this is trying to take our traditional Harlan and DeWitt germplasm classification. So our primary germplasm, which are everything that is crossable without any crossing barriers at all. Our secondary germplasm that has some meiotic abnormalities, but crossing is still relatively easy. And our tertiary germplasm, where crossing is possible, but you need to use a specialized technique like embryo rescue or something like that. And choosing and taking the evolutionary relationships to pick which crop wild relatives might be the best for crossing. Now, this is very good. You can find species from that live in extreme climatic niches or extreme places, and that are likely to cross well. However, the problem with this method is it tells you nothing about an accession. It just tells you which species from an extreme climate is likely to be useful. And the last method that's become really popular is landscape genomics. And this is where you use an association mapping approach, so a GWAS approach, but instead of using a phenotype, you use the environmental data from the location where the sample was collected. Now, this method relies upon it being a landrace or a wild species because the assumption is that the geographic location, that natural selection has acted for many generations on that specific line or population, and that assumption we feel can be fairly made in a wild species or a landrace. It's not fair to make that assumption in modern breeding programs where a lot of the linkages between place and that the long-term selection may have been broken. This method has some issues in that again, we don't necessarily get to an accession. We get an allele that is favorable for a particular climate variable. And so we get a lot of information about the potential change, but not necessarily getting down to that accession level. So what we've been really working on is how do we combine those? But before we can combine those, we're going to just look at a little bit about some previous work where we can see what you get from them. So let's look, for example, at figs. So figs, again, really popular at the turn of the century and after. This is an example from sunflower where you do the species distribution mapping of 36 different species. And what you're seeing on the right here is the black shaded area is cultivated sunflower. The red box is the primary germplasm. All the other boxes are different species in the secondary and tertiary germplasm. So what we see here is that when we look at precipitation, and when we look at soil pH, two things that matter a lot for sunflower production, we don't need to go outside the primary germplasm to find an accession that really moves this either for about twice the amount of precipitation or about half the amount of precipitation that we see in the normal rain-fed conditions. And we can go to, but there's a much more restricted range for, say, soil pH. So again, this is useful, but it's not all that useful because it just gives us these kind of broad patterns. Figs can be extended if we're looking at if we start looking at tying that to specific geographies. So, and I'm sorry, next slide. So you can take, if we look at sunflower again, and this was some great work by Zia Mehrabi, you can look at not only the current climates, but you can project it into the future. So what you can do is what you're seeing here is non-dimensionalized climate space under the current and future projections, and what you can see is that looking at crop wild relatives or looking at the materials that are grown all over the world, you can see which accessions under future conditions are actually going to be adapted to those future conditions. And what's really cool about this method, because it's this non-dimensionalized climate space, you can also project diseases and to see if they're going to overlap. Now, again, the downside here is that we're not necessarily know anything about that crossability. We just know that these accessions exist in these extreme environments. The other method that we've been doing a lot of work on next slide, is landscape genomics. So this is an example. Many of you probably know Jeff Nyhart. He did actually work in barley quite a bit. He's now the USDA cranberry breeder, but he did his PhD with Kevin Smith at University of Minnesota in the barley breeding program there. But what he did with these cranberry populations is he took all of these wild cranberry populations across the Northeast US and looked for co-localizations of environmental and soil variables across the genome. So what you're seeing in this figure is every bar is uh, the red is temperature, the blue is precipitation, the brown is soil. And you can see that there are co-localizations for these environmental variables all across the genome. And then you can pull out specific ones that, in this case, if you're looking at that Manhattan plot, there's a single QTL there that actually impacts 6 different environmental or soil variables. And so, again, this is really nice. It's really useful, but we've seen really limited use of this type of information in breeding. Actually, the only examples we have of using environmental association and breeding come from Jeff himself, who's used this data in the cranberry breeding program and in the barley breeding programs in the UK, where some of the work from Joanne Russell's group has been, there's been a series of papers that have used those. So it's very, again, fun to be talking to you guys because there's been evidence of it being used. So this is where it really comes to the method that we've been talking about here. Next slide, where what we've really been trying to do was figure out, can we combine core collections, figs, and landscape genomics? And so our inspiration was this 2016 paper from Jiming Yu's group on using genomic prediction to predict the breeding values for the gene banks, and this paper from Sam Piernan about using the climate data to look at crop replacement and what's going to work in the future. And so work that's being led by Quinn Campbell, in a PhD student in the lab, first looked at how frequently this has been used in crops, looking particularly at the treaty crops. Turns out it's not been done all that much, even though a lot of data exists. It's been done in 12 out of the 64 FAO treaty crops, but there's data for 26 of those crops available. And so it hasn't been done as much. But what we've started doing is using the core collections of germplasm, using core collections of our germplasm collections to predict the GEAVs instead of a GEBV. So a GEAV is the exact same as a GEBV, only it's an adaptive value where we're predicting those environmental tolerances rather than the than a breeding value. And the reason we really like this is now we get a specific accession that allows us to say, well, we think that this accession is going to do better for this particular environmental variable. And this leads to exactly what we think is probably going to be more interesting for you guys. It's a study that Anna led, where we did this in barley. And we had a lot of great collaborators, Sonia, who I'm sure you guys all know, and Peter Morell as well from University of Minnesota, who I'm guessing you guys know. So, and this paper should be out in 2 days if you guys want to read the whole paper. But Anna will give you a really great discussion of it now. So without further ado, Anna.
I know, I'm just making sure you can all hear me okay while we do the transition. Lovely stuff. Okay, so thank you so much for the invitation as well to speak today. So yeah, I just had the good fortune really to be involved in the conversations as in the previous slide and the papers that you saw as all of that work was coming together. And so, yeah, I was just interested in trying to apply the method that Mikey and Quinn had pioneered in another species of interest, and that just happened to be barley in this case. So yeah, the work that I'll talk about is basically the figures from this manuscript. Yeah, so the data source for this work came from this Lee et al. 2019 paper that came out of Peter Morell's group, where they had 784 barley landraces of known geographic origin, and they were genotyped. And so you can see their distribution here in Eurasia on the right. And the original publication was looking at FST outliers to identify key genes for temperature and drought tolerance. And the samples stratified into these 5 main population groups that you see here, and on the PCA on the left. And so one of the things that was apparent fairly early on was that there seemed to be a relationship between these population divisions and the various bioclimatic variables, or in this case, the world climate variables that you see here. So just as an example, on the left, we see population 2 here showing a relationship with bio-14 and bio-17. These are precipitation variables. So we felt this data set was a good fit to bring the method of environmental genomic selection to. So as EGS is an extension of GS, as Mikey was saying, the classic question of what is the appropriate training and test divisions arises. So here we evaluated a very small core of 31, which represented the overlap between the USDA's mini core that came out of this 2014 paper and the 784 sessions from the Lee paper. And then we also generated a de novo core of 100 using Core Hunter to try to compare just between these two. And so the graph here just shows the prediction accuracies for the WorldClim bioclimatic traits. And we tested 4 different GS models. So yeah, if you're familiar with GS, in this case, the approach is similar in that where we would traditionally have a genomic estimated breeding value for an individual for any particular trait. In this case, we're substituting the WorldClim bioclimatic variables as the phenotype for our landraces, and then conceptualizing the results as genomic estimated adaptive values, or GAVs, for any given trait. And so the red boxes are just the ones that we focused on. I'll focus on more in some downstream figures here. But the takeaway from this is basically that the core of 31 actually held up quite well in comparison to the core of 100, which is interesting. So yeah, as I was kind of curious really about how these various populations or haplogroups might differ in their means for in these GAVs for the different traits or environmental traits. And so we grouped the results by these population divisions, and it was just interesting to see how the groups differ across these variables. So to save your eyes a bit, I'll just zoom in on 2. So for bio 1 here, the annual mean temperature, we can see that population 1 and 3 have a higher GAV mean for this climate variable versus, say, another variable like bio 4 temperature seasonality, where population 5 shows a very clear advantage in this scenario versus the other populations. And just taking it a little step further, it's also interesting to visualize the individual marker effects, as the method works similarly to GS by assigning individual marker effects to every SNP and then summing over those for the breeding value for the trait. So yes, barley has 7 chromosomes, but just to save your eyes, I've just plotted 3 here to show you. And this is for bio 1 across the 5 variables. So blue in this case indicates a positive marker effect, and red is a negative marker effect. And the thing that's important just to mention here as well is that our overall chromosome coverage is pretty poor. We only had about 6,000 markers altogether. And so the black lines that you might be able to see here represent 1 million base pair jumps, and so we can't draw any conclusions about, say, like the proximal arm of chromosome 1 has a more negative marker effect, say, directly. So it was just shrunk here just to fit the space, really, just for the ease of display for the plot. So nonetheless, I'll just zoom in here on a section of chromosome 1 of population 2. And so you can see a little bit more clearly, hopefully, the patterns across the samples, say within a population, where the marker effects align in their positive or their negative associations at specific loci. So it's just a fun way to think about visualizing the markers for a complex trait and to understand how these patterns might differ across samples or across populations or between populations. Alternatively, we can also visualize each sample's overall GAV value in physical space. So here, sessions are colored by their GAVs, where I'm kind of testing if you're paying attention here, I suppose, because I've switched that red is actually positive in this case and blue is negative. So we can see that, say, the African examples, as you know, have a high GAV value for bio1, say, or annual mean temperature in this case. And we can obviously do this for any trait. So in this example, then, these are the breeding values for bio4 or for temperature seasonality plotted, where the African samples naturally show a poor breeding value for seasonality, whereas in more temperate areas of Eurasia, we see a higher breeding value for seasonality. I was also interested in trying to bring together EGS with other climate modeling techniques, in this case, species distribution modeling. And more specifically, for when we're thinking of methods or approaches for parental selection in breeding. So I ran population level species distribution models, and the plot here shows those different distribution models for each of the 5 populations. I'm looking more specifically at an example here. This is the SDM result in red for population 1, and overlaid on top of that then is the GAV values for all of the 784 samples. So rather than having to consider all 784 lines, if we remove those lines for ones that do not match the geographical species distribution of population 1, it restricts our potential selections down to those that have a high GAV value, whilst considering the climate that your breeding program may be located in. So yeah, just a way to kind of bring together EGS and SDM. And obviously you can do this with each population. So in this case, this is population 2 in a temperate environment, so, or temperate Eurasia, where when it comes to your parental selections, you have a lot more availability if you're in a temperate region versus a much more tough region growing in Africa. Yeah, if you're interested, all of the GAV values and the ranks and all that stuff for the lines are available in supplemental tables of the manuscript. We had attempted to suggest that the approaches in general here may avoid linkage drag when you're making parental selections, so using the 2 methods combined. However, a reviewer did very rightly point out that we did not test this. So next steps would include some validation methods, most certainly. And other things that we're thinking of, just here in the plots. So this is data for another species, where we saw changing GAVs over a 12-month period. So this is actually the months of the year on the x-axis, with a specific climate class, in this case, corresponding with monsoon season. So we've gone a little further and investigating candidate markers that are kind of dragging up this mean in the GAVs that you see here. And we hope to bioarchive this work within the next week or so, if you're curious. And yeah, just I guess thank you so much for your attention. Obviously, none of the work is done alone. It's a huge group of people, and I'm very grateful for being included in all the conversations and the development, and I hope you enjoyed the talk. Thank you.
Thanks very much.
Yeah, thank you.
And Michael, that's great. So I think we open the floor now to if anybody has any questions. But maybe I could start. I can ask one to get us going. Just about what, like, you bring in data together from all different places in this work, and just like what sort of challenges and issues are you having, and how do you resolve those and that kind of thing?
So I think it's something that we've actually tried to think about a lot, because all of the data issues are really important, because it's really hard to generate data. And so one of the things that we've really tried to do a lot is work with people who were the primary data generators in a lot of these areas. So, you know, for example, in the work here, you know, working with Peter on this paper. But I think more generally, we're really in favor of open data, because the value that we see in a lot of this work is having the plants. You know, the ability to play with the methods and see what we can learn from all of the data that's generated. And Paul, like we were talking about slightly before, what's great is The barley community has been great. The metadata are really good. The data are easy to follow. They're easy to use. So it lets you do a lot of this. It a lot to try and understand how these methods are going to work. And so it can give us this kind of intuition. And one of the things that we've really been kind of thinking about is how many populations can you reasonably assess if you want to make all of the crosses, right? Oh, sorry. But yeah, but so for us, it popped up in the chat the question, then left really soon. But for us, we really think we want to make everything as open as possible so anyone can use it, because we want the method to be species agnostic, and then all the experts in it to be able to take the information that we generate. I should also give a pitch for Anna's GitHub, which is really nice. If you guys want to do the complete analysis, all the scripts are up there, all the data is up there, and we're happy to help try and explain any of that. But for us, the way that we think about it is we want these gene banks to be libraries, and the best way to make them libraries is to kind of give the full recipe for these analyses so that anyone can use them for whatever use that they want, if that makes sense. Hopefully that answers the question.
Great. So another question from Robbie Walsh here in the IBH here. So this is at Anna. In the barley work for the analysis, did you partition the populations according to row type or growth habit? And can you normalize for these major historical genetic discriminators in your analysis?
I didn't. Could I? Probably, most certainly. We, as far as I know, they're all 6-row, but I could be wrong. Yeah, that's a really fun idea. But no, I didn't in this case. Everyone was treated the same, and there was no, other than the population stratification. I didn't change the analysis for anyone in any particular way.
Okay. Another question from Robbie said, not sure if you can see or hear him. Do you want to try and talk, Robbie, or?
Can't figure out where the chat is.
It's maybe we can go into another question just now until it happens. So
Okay.
Okay, Robbie can't mute himself, which is you'll not be able to speak. But another question. So we've got one here from Sebastian Raubach, who works with me here in Dundee. Do you have an issue with gene bank mix-ups in your work, and how do you deal with that?
So Peter has actually dealt with that extensively, and so luckily that work had been done prior to us taking them. The best way that we can figure to deal with it is to look at your pairwise marker identity once you have everything genotyped, and eliminate things that are identical or near identical. Because that's not going to provide you any additional information. That is obviously less of an issue now because of computational power. It's not hard to do those comparisons. 20 years ago that was really hard when I first started doing those, right? But now it's fairly trivial. But it's this is a big issue in all the collections, right? What I've heard, what a lot of the gene bank leaders talk about, right sizing their collections by genotyping everything and doing those studies. And again, like IPK has done a great job of doing that and kind of leading the way in that.
Can I put you on the spot? What percentage of gene bank accesses do you think are not what they say they are?
So depends on the species. So Anna's laughing because we have another project where we've been working on this actually in Hawaiian heritage varieties of sweet potato. And so in that case, it's a really high percentage where the names are not correct. I would say probably in that collection about 20% are incorrect. Where the names don't match where the plants that you grow from the name do not match historical descriptions or previous or historical botanical descriptions of what used to have been grown. For the seed plants, not clonally propagated ones that have been maintained in living collections, it's much, much lower. So for the seed collections, things appear to be, it's much more like I think like 1 or 2% of things that are actually incorrect. And it's not actually incorrect. It's just multiple. Typically, what I've seen in the other collections is it's not things that are misnamed. It's you have multiple accessions that are the same name. And so that's a relatively small percentage. And a lot of that just has to do with so actually I blame breeders. Because breeders are great at sharing seed, and they share things at like an at an F4 or something like that, and so you get these things that are kind of different but not really different, and very very identical with maybe slightly different, like some heterogeneity around introgressions for particular traits. But then like when a breeder retires or something, they'll donate their entire collection to a national gene bank, and they have their own special code for it, or a shorthand in their notebooks, that makes it seem like it's unique when it's really something that they got from their friend.
Yeah, I think duplicate names, different names for the same thing is a problem we've always got to deal with.
Yeah.
So we have another question here from SB. Thank you for the excellent talk. Were you able to capture structural variations in your analyses? If not, what strategies could you use to incorporate this layer of genetic diversity?
So no, we weren't able to get structural variants. I think, and sorry for interrupting, Anna, but I think we would need to do whole genome sequencing. I'm not particularly, and maybe you guys have had other experiences, but so this was actually array data. So SNP array data. So the genotyping was quite good, but none of the things there were actually tagged for the structural variants. I am not personally particularly convinced of calling structural variants with GBS data. I've had really mixed results with that, where sometimes I'm like, oh, this is amazing, and then I try it on a different project and it doesn't work very well. But for the whole genome sequence, it seems really, really good. And I actually think that would be a great thing to do, because I would guess that there's quite a number of kind of mini inversions that are protecting some of these environmentally adaptive regions, because we see that in the evolutionary biology literature. We see that quite a bit, of these kind of smallish structural variation, particularly inversions, that really matter. But we weren't able to detect them. But we would love to try.
Okay, we've got another question for Robbie Wach. I don't know, can you speak, Robbie, yet, or are you still muted?
Oh, can you hear me now?
Yes.
Yeah.
Oh, unbelievable. Something just happened. That's the first time there's little video buttons popped up in the bottom of my screen, and now I can unmute. Perfect. So yeah, both of you spoke quite a lot about, well, you spoke exclusively about landraces and the value of the genetic material that's present in landraces. But what about wild germplasm? Because the biggest bottleneck, I think you showed in your very first slide, the Tanksley and McCooch thing, was that the biggest bottleneck was from the wild to the cultivated line. So how do we exploit the wild material? How would you do it?
Do you want to go, Anna, or I can take a swing?
I mean, I would compare, I guess, like I would think about, like, wild most certainly, right, are going to have probably a lot stronger association with the environment than a landrace, right, that we've selected, and that's kind of we've kind of babied it a little bit more than we have maybe a wild, right? So the only problem with that,
I would disagree there.
Oh, okay.
First of all, I
That's
disagree
okay.
because very often the wild material comes from a very restricted geographic domain.
Yeah.
So it's not
Okay.
really been exposed to an awful lot of the environmental variation that you've included in the landrace
Yeah.
material. It's not really had the chance to adapt to that different environmental variables. Anyway, sorry. Sorry,
No.
just as a discussion rather
No,
than a
no, no, no,
question.
no, no, no. I
No,
think
it's
it's
good.
really,
It's good.
I think it's really interesting. So I think for the wild material, if you have good collections, so like some of the collections from, some of the wild collections, from the Middle East, I think one of the things that's a bit tricky with some of the wild populations is they do sometimes end up being yeah, kind of dogs that end up being in kind of unique microenvironments. Where so like I know like you get some of the collection trips on like the Israel Jordan border. I know that you get some populations where there's almost zero precipitation, but because they end up being in these, close to the Jordan River, they end up actually being okay. But I think there's it should work pretty well with all the wild species. Where I think the thing that's harder with the wild species would be the length of time that it would take to do those introgressions is obviously going to be quite a bit longer. And I think a better strategy for if you're actually doing the wild species would not be this environmental genomic selection or environmental genomic prediction that we're looking at, but actually going back and doing what Jeff had done with the environmental GWAS, and then actually looking for any of those alleles, and then um, then genotyping for those alleles or those genes in the germplasm for something that actually is in the collection that's slightly closer to what you're cultivating. So I think it'd be a slightly different process to leverage those as well, because I think it's much. I mean, I guess just thinking back to the sunflowers, I'd much rather work with a landrace than the wild species. And so I think it'd be easier to do that. But it does work pretty well with the wild individuals as well. You just need to make sure you have good um, collection data and good GPS coordinates from where you've done the collection. Hopefully that helps. But yeah, but it does, and one of the things that you do see signals of some of the kind of the famous genes. And so that's, some of the work that Peter did before, where you found a lot of kind of the well-known genes associated with the different environments, or the different places where you grow it in some of those lines.
Thank you.
Yeah.
Great. We have one more question here from Luke Ramsey, who's also based here in Magowrie.
Awesome.
Anna, you mentioned avoiding linkage drag in the utilization of results. Could you expand?
Yeah. I mean, the reviewer was totally right. We didn't really actively assess it. My rationale there is like, well, if we're breeding, within a haplogroup, then, we would reduce this potential linkage drag by breeding individuals that are more similarly related to each other versus, populations that might be in very different geographic locations or environment classes and things. I mean, a perfect way would really to be just to do exactly that, and then in the progeny, figure out, what the effect on that linkage drag truly is. The validation is really the only way to prove that point. And yeah, if we could get funding, we could do it. That would be fantastic. We're certainly thinking about like grant applications and things for the future. So yeah, I think a validation really would strengthen the argument and hopefully it's the hypothesis is correct. We won't know until we test it.
Yeah, and I guess one thing that I'll add is one of the things that we've been thinking about in terms of this is just trying to reduce the number of parents and the number of crosses that you want to make. So we kind of back calculated what's the highest number of populations that we here in Hawaii could evaluate in a given year or a given season, and then kind of back calculate that to how long would it take if that's how many populations you evaluated it in a year, and you wanted to do every single pairwise cross in an entire collection, right? So if you want to do every single pairwise cross in the even amongst the 784 land races that you have here, right, that would take us about 30 years to do those evaluations here, which just isn't feasible. So we want to get it, the way we kind of think about it is, how can we get it down to a number of parents that we're reasonably sure, kind of stacking the deck in our favor, so that we can evaluate, instead of trying to evaluate, 50,000 populations, or 100,000 populations, can we evaluate, 500 populations or 100 populations, get it down to a tractable level where we're not so scared by the numbers that we kind of throw our hands up and say that this is not a reasonable experiment to try and design.
Great. So I see Robbie's made an appearance in person now. I think
Nice.
Do you need a couple of other questions, Robbie, or another question?
Well, I didn't want to dominate the question. So no,
We're happy to answer more.
Well, I think you probably know already anyway, but we have particular interest in the wild germplasm. We've worked on the land races in the past. I think I guess it's a developing thought in that there's an awful lot of valuable, probably more valuable variation in the land races than there are in wild germplasm, particularly because of a lot of the variation in the wild germplasms through rare alleles, which you wouldn't detect using environmental GWAS anyway.
Yeah, no, you wouldn't.
Its geography is restricted. Very often, well, certainly if I talk about barley and wheat, it's restricted to the Near East Fertile Crescent for the wild species or wild relatives.
Yeah.
And I guess you started to address the question in a sense that it becomes a very intractable question or problem when you think about looking at, making biparental crosses between hundreds of individuals with multiple parents. We've done some stuff with a dozen wild species or a dozen wild individuals.
That's so cool. Yeah.
And if Ruth's online, she might want to comment on this, but she's already up to evaluating thousands and thousands of individuals with 12 parents. And it just becomes I say almost intractable. The one I guess shining light on the horizon might be some work that was conducted by Scott Bowden and Song Koo in Adelaide, where they made crosses between wild species and cultivated species. And I think if I recollect correctly, that they could predict the best performing wild species lines, in other words, the lines that have got positive genetic variation in the F1 generation.
Oh, that's awesome.
Which I thought was really quite good, because it means that your population size is 1 to evaluate an accession if it translates into that in the wild species. But who knows? And I don't know if it actually would happen also for land races, if you could see advantage in the F1. So it's maybe something to think about, an alternative strategy.
No, but I think that's exactly right. Like because it's so much of the way that at least that I think about it is a brute force approach, of evaluating everything is not possible, right? So we need to use, and I think this is where we can use a lot of these techniques to try and figure out, okay, how do we stack the deck in our favor? How do we pick the best potential, our best potential winners? And even if we're not, even if we're only kind of right, it still lets us do it. It actually lets us design an experiment that will be better than what we have without, without and it'll be much better than random, right? Which is in a lot of cases, which is what we want. Can we do better?
I mean, I think if you can do things very early in very early generations on small
Yeah.
numbers of individuals, things become
Agree.
really nice to see you were working with the people in Minneapolis, by the way. I'm guessing they gave some valuable advice.
Oh, definitely. Definitely. And I should say, I do remember you gave a talk in Minnesota in about 2011, I think it was, but you were visiting Gary. So
And are you both in Hawaii just now, or Anna, are you
Yep.
in Ireland?
no,
You're both
Oh,
in Hawaii. So
I'm
it's almost
listening to
bedtime.
America, yeah. Oh yeah, it's well past bedtime.
Yeah, listen, thanks so much for
Oh,
taking the time, especially so
Oh,
late
thank
at night
you.
to talk to us all.
thanks for the invite. It's
Oh, thank you.
it's really fun, and I don't know if Sonia's here, but thanks for all your help with the paper, Sonia.
No, I'll just say again, thanks so much for the presentation. It's really good, just at the right level, and I'm
Thanks.
hoping that everybody else online enjoyed them as much as I did. So
Yeah.
take care.
Thank you.
Lovely
Thank
to
you
see
so much.
And hope you can join future talks. Even though
Awesome.
they'll be
Thank
late
you.
at night.
Thank you.
See you later.
Thank you.
Bye-bye.
See you later. Bye. I saw one more question.
Yeah, there's one more question here from Luke.
Oh,
Thanks.
Steve.
The approach is evidently focused on adaptation to abiotic-biotic stresses. Is there any scope to screen for quality human use traits?
Ah.
So I think in that case, you could use a similar approach to what we did, because that's what the Jimming U paper did in 2016, where they predicted the entire sorghum collection for all of the for all of the phenotypes using the core collection with quite a similar level of success in terms of having quite high prediction accuracies, the same way that we did. So I mean, like I said, that's where we got the inspiration for using the core collections. And one of the things I know Anna mentioned it, that I was super excited about, was that when Anna just developed her own core collection from everything that was genotyped, those prediction accuracies were really high, right? So even if it's not the like the traditional core collection, even if you don't have an overlap, if you do that same kind of stratified sampling for your for the initial for that initial prediction among everything that's genotyped, it will work very well. One thing that we haven't done that we're quite keen to try and do is figure out when you would, like, after how many generations of selection do you have to get your new training population. And we haven't gotten around to doing that yet with the germplasm, but I know Anne and I've been talking about some ways, and maybe you guys have some really good thoughts on it, but maybe thinking about some ways of doing maybe some simulation work to figure out how frequently you'd have to retrain and what that loss in accuracy would be after doing this. But yeah, but I really highly recommend that Jimming U paper from 2016, because it should work for all of those, for all the human use traits.
Great, thanks.
Yeah.
So if there are no more questions, just like to thank the speakers again for the fantastic talk today. Thanks everybody for coming along, taking the time out of their day to come and listen as well. Just encourage people to keep an eye out on emails and the Barley Hub website for future events. And I think also this is recorded, so if people want to listen to it again, maybe have some more questions for Michael and Anna, they can do so at their leisure and get back to you. So until then, I guess bye-bye for now, and look forward to seeing you at a future event.
Thanks so much, both of you.
Thank you.
Thank you.
Bye.
Thank you. Aloha.
And